Glyxon Biolabs. How may I direct your call?
Hello, my name is Jane Stafford. I'm a journalist with the Global Scientist. I'm calling to inquire about the possibility of interviewing some of your lab members about your exciting new project.
Yes, the exciting new project. And what publication did you say you were with again?
The Global Scientist, we're a well respected publication with a focus on scientific breakthroughs and the implications for the future of humanity.
Can you hold, please?
Hello, this is Dr. McClintock, Head of Research. I understand you're interested in our project.
Dr. McClintock, yes, that's right. I'm very interested in learning more about it. It sounds groundbreaking.
Oh, well, that's very generous. I wouldn't say groundbreaking. We're just scratching the surface. But predicting microRNA/DNA interactions is crucial for advancing plant biotechnology to a new level of refinement and precision. Plants rely on intricate gene regulatory networks for growth and responses. MicroRNAs fine-tune gene expression with exquisite specificity. So, I think I should introduce you to my colleague, Victor Ambrose. He is our expert in microRNA. He is an authority in the field. You can ask him more details.
Thank you, Dr. McClintock.
Victor, do you have a minute? Is Jane Stafford with the Global Scientist? She wants to know more about our exiting project here at Glyxon.
Hello, Jane. I'm glad that you want to find out more about our research.
Nice to meet you, Dr. Ambrose.
The goal of this project is to, firstly, develop a model that can help us to understand the complexity of microRNAs and microDNAs as signaling morphogenetic molecules while targeting a specific group of genes across plants' genomes. Those genes are suspected candidates known for controlling multiple morphological traits. In order to achieve that map road of interactions, we have incorporated the use of machine learning algorithms. One of them works, searching the functionalities of a suspected microRNAs. Another, program assists us to localize the interacting species between other microRNAs or pieces of a gene. Using generative AI algorithms help us to save time analyzing thousands of hundreds of candidates by narrowing down those with the best potential interactions, but also to design microRNA probes more accurately that we suspect could be used to downregulate or upregulate morphogenetic traits. The best candidates of the microRNA are synthesized and tested in vitro and in vivo conditions and then tracked down using fluorescent beacons. Our goal is to automate the process using optogenetic devices in growth vessels and chambers for sequentially controlling the expression of microRNA in engineered plants.
Oh, wow. That sounds incredibly complicated. I have a couple questions, Dr. Ambrose. In layman's terms, how does your research addresses the must-urgent challenges in climate change, food security, and biodiversity conservation? And were we able to see soon its implementation in real-life scenarios?
You know, Jane, working with microRNAs is not just a technical challenge per se. Its outcomes could revolutionize plant biotechnology, regenerative medicine, biomaterials, and biodiversity conservation. But regarding the plant models we study, we can argue synthetic microRNAs is a very promising technology that can help to improve crops at a much faster rate, and it's tremendously beneficial to restore endangered species that are difficult to propagate. Well, and I think our senior advisor, Dr. Carver, could contribute with a broader overview on the agricultural implications of our research. Hey, George. Could you help us here?
Hi, Jane. I couldn't avoid listening your questions. I believe the key is about communication. We know very little about how cells differentiate and become complex multicellular organisms with organs and tissues, with specialized functions and metabolisms. So Dr. Ambrose's approach is possibly the most elegant missing link connecting development and the environmental influence on plants' development. I bet microRNAs could also offer clues on understanding the origin of multicellularity. Plant cells are remarkable in their ability to communicate with each other through signaling molecules. One such molecule, RNA or microRNAs, is exported from certain cells, triggering changes in neighboring cells. And the entire process is repeated within the Leipzig cell. This intricate communication system allows plants to respond to environment and adapt to various challenging conditions, such as drought, flooding, pathogens, thermal and mechanical stress, to mention a few. Oh, this is a fascinating process. These RNA molecules act as signaling morphogens capable of reshaping the structure of leaves, stalks, flowers, and even trigger physiological responses to environmental cues.
Predicting interactions helps map these networks, understand gene connections, and identify key manipulation points. By knowing which microRNAs regulate specific genes, we can modify plant traits more accurately. Strategies include overexpressing or blocking microRNAs and engineering synthetic microRNAs for accuracy. This knowledge enhances crop improvement in yield, quality, disease resistance, and climate resilience. Predictive tools speed up the identification of microRNA/DNA interactions, leading to faster innovation in biotechnology. In essence, predicting these interactions provides tools for understanding plant genetics and creating sustainable agriculture solutions.
One last question. Do you think we could see in the near future an integration of the dynamics of microRNAs diffusion in their morphogenetic influence in a simulation or a mathematical model so we can choose from an app the shape of a plant?
I guess Victor is better equipped to answer this question. Victor?
I'm happy you asked about this, Jane. Mathematical models about plant morphogens exist since the early years of the 20th century. For instance, it was Alan Turing who developed the theoretical work about the chemical basis of morphogenesis using differential equations. His work led to what is known now as reaction diffusion models, presuming an asymmetrical diffusion patterns of morphogens. Like a teabag infusing in hot water. Very British of him. Louis Wolpert also proposed the French flag model that works similarly. Nevertheless, those models aren't always infallible to explain morphogens' activity, so experimental work is still needed. We propose that the Argonaut system have a more important role now. In present time, researchers like Przemysław Prusienkiewicz and Masayoshi Kawaguchi have demonstrated the existence of reaction diffusion activity of plant morphogens in apical meristems, but for very specific signals. Sadly, the Jetsons Plants boutique is years from now, but we are closer.
This is fascinating. I'm so glad I contacted Glyxon Biolabs. Your research is intriguing, challenging, and inspiring. I appreciate the time you share with me, dissipating my doubts. I appreciate it. I can't wait to hear more of his future advances. I wish you all the best.
Thank you so much, Jane.
We are happy to share our journey.
We are humbled. Thank you.